Hello, my name is Paul Howes and I'm the manager for the Centre for Sustainable Aquatic Research, CSAR.
And I'm Sara Brento. We are based at Swansea University and CSAR is a centre of excellence. We do research on aquatic organisms, including microalgae and seaweed.
We have a number of different fish species within the facilities. We have salmon, we have tilapia, we have killifish, we have zebrafish, and we also have lumpfish.
We are particularly interested in sustainable aquaculture and conservation.
CSAR have a range of different focus areas. We focus on fish nutrition and fish health. We also focus on fish welfare. We have a number of links with industry and we try to develop the sector within Wales as much as possible.
The Access to Sea project, for instance, focuses on supporting local businesses, especially in aquaculture, and also it aims to improve the social acceptability of aquaculture.
In the UK, aquaculture is synonymous with salmon farming. This is due to its high volume and high value rate.
But salmon and trout farms, they are targeted by welfare advocates, and many different NGO campaigns show fish as intelligent and sentient animals, and rightly so.
Within CSAR, one of our largest research groups focuses on fish welfare. We work closely with the RSPCA, a range of salmon companies and aquaculture producers to improve the welfare of farmed fish within UK waters.
But the best person to show us and talk about fish welfare is without doubt Carolina.
Hi, my name is Carolina Gutierrez and I am a fish vet. I am now working on the last year of my PhD, working with health and welfare of the lumpfish. Lumpfish are farmed and deployed into commercial salmon farms to control a parasite, the sea lice. They are also called cleaner fish because they clean the parasite of the salmon. My aim as a fish vet is to take care of and also make sure that all the lumpfish are healthy and they are reared with outstanding husbandry practices based on sound welfare guidelines. There are welfare standards for a few farmed fish, such as Atlantic salmon, but for lumpfish this is still missing. My work is to develop, along with my colleagues and my supervisors, welfare standards that are specific for lumpfish. CSAR has very good collaborations with salmon companies, lumpfish producers, and also welfare standard organizations. CSAR has outstanding facilities to rear lumpfish from egg to juveniles, and they have produced more than 3 million that has helped develop the industry. Here at Swansea University, we have dedicated labs to do genetics and to study the lumpfish microbiome. The microbiome meets all the genes from microbes living in lumpfish, and it's super important to understand lumpfish health. I am really enjoying my PhD, and I had the opportunity to travel to the most remote salmon farms up in Scotland to sample the lumpfish from the sea cages. And I have talked with fish technicians, with managers, and I have also networked with welfare researchers. I had also the opportunity to be part of the first symposium on welfare in aquaculture, which was organized by CSAR last year. And now we are getting ready to deliver the second symposium on welfare in aquaculture this November. I am looking forward to seeing my work and the welfare standards implemented in the farms.
With the Access to Sea support, we are working with fish farmers and welfare organizations to implement these welfare standards in a practical way that makes sense not only to the fish, to the farmers, but also to the consumers.
Another key aspect of fish welfare is fish health and fish nutrition. We are developing a range of microalgae-based diets to improve fish health.
Tamzin and Moritz can guide us through in terms of what to do in fish health studies and diet at CSAR.
So we investigate the effects of different environmental factors on the health of fish.
The factors we study include things like diet, environment, temperature and stress.
This is really important because we need to improve the health and the welfare of fish in farming.
This can help us to understand the changes that occur to wild populations of fish.
In CSAR, we work on several different types of fish. These include salmon and tilapia, and also killifish and the zebrafish.
We have different facilities for individual fish species to keep them to their requirements.
At the moment, we're trying to investigate how diet affects the health of zebrafish. We're really interested in finding out how oil from microalgae can improve the health of zebrafish. Zebrafish are a really good model species because they've got a well-characterised genome, they grow really, really fast, and they produce lots of eggs every single day. The eggs are transparent, which means we can see inside the eggs and see exactly what's happening with the developing embryo.
Our studies on zebrafish will also help to extrapolate these findings to other species. To study fish health, we need the aquaria to maintain the fish, and they have different size for different fish species, and we need specialized lab equipment to study the molecular level, what's going on inside the cells.
We look at changes in the microbiome. So each of these fish, just like humans, has got trillions of different microbes living in their gut. We also look at changes in the genes of these fish. We look at their gene expression and how the genes are regulated and how these are affected by differences in the environment.
Together, all this information will help us to understand how these environmental factors are influencing whitefish populations and how we can improve fish health in the wild and in captivity.
But our work is not only focusing on fish, we also grow microalgae and seaweed. We have custom-built bioreactors for growing microalgae. And for instance, with the support of the Access to Sea project, we are collaborating with local community benefit societies and other companies who are now interested to start growing seaweeds commercially and for marketing purposes.
At CSAR, we are also interested in conservation and aquatic invasive species. We work with a range of different techniques, including environmental eDNA. Jess has been doing some exciting work out in the Falkland Islands with brown trout.
My name is Jess and I'm working on invasive brown trout in the Falkland. Brown trout were introduced to the Falkland Islands in the 1940s and 50s. Since their introduction, they have spread throughout the islands and caused a reduction in the abundance and distribution of the native galactids through predation and competition. My aim is to determine the current distribution of brown trout and potential refuge for native galactids. Environmental DNA, or eDNA, is a method that allows us to detect the presence or absence of a species based on the DNA present in the environment. For example, fish such as brown trout will release DNA through mucus, blood or gametes into the water body. I collect water samples from rivers and ponds around the Falklands. I then filter this water to trap the DNA and then bring the filters back to Swansea. In Swansea, I extract this DNA in a dedicated sterile eDNA lab. Then using this DNA and specific reactions, I am able to determine what species was present in the water body. This information can help us determine the current distribution and spread of brown trout and establish refuges for native zebra trout, which can help us protect the species from extinction in the Falklands. I really enjoy my PhD as it's given me the opportunity to work with so many amazing people and travel 8,000 miles away to sample some fish in some amazing rivers. I've been able to learn many new lab and field skills and have the opportunity to present at conferences here in the UK and in Spain.
My name's Paul Howes. I'm the manager for the Centre for Sustainable Aquatic Research. I manage the facilities. We have around 15 different recirculating aquaculture systems and other aquatic research laboratories. I thought it would be a great opportunity today for you to come and see each of the different rooms that we have here. We have recirculating systems that have a range from being very small practical systems that we can use on a day-to-day basis for smaller, shorter projects, all the way up to industry scale aquatic recirculating systems that are up to around 60 cubic meters in size. Welcome to the first room in CSAR. This is the largest room that we have within the facilities. So this is the room RAS B, so it's a recirculating aquaculture system B. This is where we do most of our commercial work in terms of large numbers of fish, large numbers of production, high densities of tanks. This room has 12 1,500 litre tanks and 9 3 cubic metre tanks. This allows us to have many fish being able to be grown within these systems, which allows us to compare any of the results or the experiments that we're doing with real commercial levels and numbers of fish. So as you can see within these systems currently, the tanks are in the stage of just being cleaned, ready for the new system. And now we have different batches of lumpfish eggs incubating in these incubators. Within this particular system, over the last 4 or 5 years, we've produced close to 3 million lumpfish within these facilities, within this one particular system. Many of them very small, but some of them larger deployment size fish which get sent up to the salmon cages in Scotland. This room is one of our new rooms that have been funded through the SMARTAQUA project. This particular room is a twin system recirculating aquaculture system. As you can see, there's 12 tanks on either system. This allows us to modify or change either the salinity, the pH, or the turbidity, or the flow rates within the systems, so we can fine-tune how the fish respond to different parameters that they may be exposed to, and how that would therefore affect things like feed conversion rates and the growth of the fish as well. This is our ornamentals lab. The aim is to have a range of different marine ornamentals within these systems, where we're going to be looking at different feeding types, different feed trials, and how we can boost and benefit the welfare and the quality of the fish that are being raised within the ornamentals industry in the UK. We'll be looking at how different parameters can affect the colour and the quality of the fish that are being produced within the marine ornamental industry. Now we're entering the CRISPR laboratory. This is a laboratory where we focus on gene editing. We have a range of different species that we work with here. All of them are animal models, such as the zebrafish. We also have killifish, which are able to clone themselves. And we also are about to acquire some turquoise killifish as well. We look at a range of different studies here, so studies to do with longevity and aging, studies to do with obesity, studies to do with diet requirements, and these species can act as a model for similar mechanisms that would happen within humans. Okay, now we're moving to some of the medium-sized systems that we have within CSAR. Within this laboratory, we look at how different trophic levels interact with each other, and the main aim of working with the aquaponics area here is that we have a range of species of fish we can hold in tanks or filter feeders, and we can also look at how the waste from these particular species can grow different plants. Okay, so we're now entering C2. This is a room that we use for innovation, so it's our innovations lab. We work with a range of different industry partners within this room, as well as a number of different students who do short trials in order to answer questions for industry. You can see that we have a range of different tanks. They're all in multiple numbers, so lots of duplicates and replications for getting decent quality statistical data out the other end of the trial. Okay, so now we're entering a lab that looks quite different to some of the other labs we've been in previously. These particular laboratories are designed specifically for model organisms again, so zebrafish this time. As you can see, they're in a rack formation with multiple small tanks. This again allows for a lot of replication. Within this room, we're concentrating on improving and standardizing the welfare of these particular species that are now one of the most commonly used organisms within animal research for model organisms for humans. This particular room is C4. This is the microalgae lab. Within this laboratory, we hold our main master cultures. So we have 25 plus different master cultures, some of which we've had for 30, 40, 50 years. Each week they need to be re-inoculated, and from these master cultures we can increase the size quite rapidly through to small beakers, then to carboids. From carboids they can go into these bags, and from the bags we can move them out to the biofences that you'll see later on within the facilities. So we're now entering tropical lab. As you can see, the temperatures are really quite high. It's kept at around 28 degrees water, and the air temperature is around 25 degrees as well. So it's a pleasant place to work during wintertime especially. Within the tropical lab, the system is set up to have the full life cycle of tropical species. So we have broodstock tanks here, we have the larval tanks, we have the fry tanks, and it's all controlled again by a main recirculating aquaculture system. So this particular building here is a building that's being created specifically to look at the different trophic levels within aquaculture and how they interact with each other. The unique and novel part about this building is that we have a glass house built onto the end of these two main recirculating aquaculture systems, and within the glass house we have a PBR which allows us to grow microalgae based on the nutrients that are produced from the waste from the fish. The main aim of this is to try and look at ways to capture opportunities within the circular economy. That is, by which we can have waste streams and make the waste streams themselves a way to generate new income or new products within the circular economy.
So to record the welfare of the lumpfish, we are going to start by taking the weight. So we weigh the fish very carefully. And then we are going to measure it. If the tail is missing, then we are going to measure till the longest point. 76, and we record it. So to assess the welfare of the fish, we are going to now measure 5 different parameters that we are going to score from 0